This might shock you, but Earth is in the middle of a mass extinction event right now. More than 1 in 5 species faces extinction, and scientists warn that that could rise to 50% by the end of the century. But what if extinction wasn't forever? Thanks to breakthroughs in genetics, cloning, and biotechnology, we may be able to bring some of these species back. Today, we'll explore the history of extinction and the innovative science reshaping its future. So why do animals go extinct? Well, extinction has many causes, but most of them are linked to human activity. First up is overhunting and persecution. The moa of New Zealand was wiped out by Maori hunters just 600 years ago. The dodo was slaughtered by sailors in the 1600s. The thylacine, or Tasmanian tiger, was hunted under government bounties, with the last dying in a zoo in 1936. And finally, passenger pigeons, once 5 billion strong, were hunted into extinction, with 250,000 birds from the last flock shot in a single day in 1896. Next up is habitat loss. Passenger pigeons lost North America's forests, while dodo's island home was destroyed by settlers. Next is invasive species. Cats, dogs, and pigs devour dodo eggs. Introduced predators continue to threaten smaller marsupials like the mountain pygmy possum. Also, there's pollution. China's Yangtze river dolphin, the beiji, went extinct in 2006 due to industrial contamination. And finally, there's climate change. Some losses predate humans. Mammoths and woolly rhinos collapsed as the Ice Age ended, while the Siberian unicorn, a giant horned rhino, couldn't adapt its picky diet to climate shifts. So how do we de-extinct an animal? For centuries, extinction was permanent, but today, scientists are developing 2 tools to reverse it. First is cloning. Cloning uses somatic cell nuclear transfer, or SCNT, to take the nucleus of a preserved cell, place it into the egg of a close living relative, and implant it into a surrogate mother. It's the process that produced Dolly the sheep in 1996, and in 2003, it briefly brought back the Pyrenean ibex. A cloned ibex was born from a goat surrogate. The first extinct animal to return, though it survived only minutes, it still proved the concept possible. Next up is gene editing. Most extinct species don't leave behind intact cells, and that's where CRISPR-Cas9 gene editing comes in. First, scientists sequence DNA from preserved remains. They then compare it with close living relatives, and then they edit out specific genes like fur thickness, immune traits, bone structure to bring back lost adaptations. Finally, they grow embryos in surrogates or in one day artificial wombs. This method doesn't make a perfect copy, but a proxy species designed to look and behave like the original. That's how scientists are working on hybrids like the mammoth, an elephant engineered with mammoth traits. So what are some future animals that scientists are hoping to bring back? Now that de-extinction is no longer just a theory, several high-profile projects are already underway. First up, the woolly mammoth. Extinct around 10,000 years ago, with some surviving 4,000 years ago on Wrangel Island. Colossal Biosciences is editing Asian elephant DNA to produce shaggy, cold-resistant calves, hopefully by 2027, aiming to restore Ice Age grasslands and slow permafrost melt. Next up, the dodo bird. Wiped out in the 1600s on an East African island of Mauritius, the genome was sequenced in 2022. Scientists are editing its closest relative, the Nicobar pigeon, though bird reproduction without wombs remains a significant hurdle. Next up, the thylacine, or the Tasmanian tiger, which went extinct in the 1930s. The genome was sequenced in 2018, and researchers are hoping to reprogram marsupial stem cells into thylacine-like embryos for artificial wombs or surrogates, reintroducing a lost predator to balance the ecosystems. Next, the moa, a giant New Zealand bird over 10 feet tall, extinct by the 1600s. The genome was reassembled in 2018, and plans call for editing moa DNA into emu eggs through avian cloning, which is still extremely difficult. Also, the passenger pigeon. Once the most abundant bird on Earth, extinct by 1900s. Revive and Restore is splicing its genes into band-tailed pigeons, aiming to rebuild flocks that could restore forest ecosystems. And finally, the Siberian unicorn, a massive Ice Age rhino with a single horn, extinct 30,000 years ago. DNA has been sequenced, but its distance from living rhinos makes revival extremely unlikely. Studying it may still help conserve modern rhinos. So why should we pursue de-extinction? Well, first, ecological restoration. Mammoths could stabilize Arctic permafrost, and thylacines could restore balance in Tasmania. Next is conservation. The same two methods are already saving endangered species, from creating embryos for the last two northern white rhinos to boosting genetic diversity in the pink pigeon. Additionally, scientific innovation. Advances in cloning and gene editing could transform agriculture, conservation, and even human medicine. And finally, righting wrongs. For species wiped out by humans, like the thylacine or dodo, de-extinction offers a chance at redemption. But it's controversial. Critics argue we should prioritize living species and warn that resurrected animals might be sickly or maladapted, and raise the ethics of playing God. What's clear is that we are in a biodiversity crisis. De-extinction may not be the silver bullet, but it's tools that could reshape conservation, giving us a way to restore ecosystems and protect what remains. The question is no longer can we bring species back, but if we should. If you enjoyed this video, please consider subscribing, and please join our community if you want early access or updates for our videos. Thanks for watching.